checkpoints where the cell is waiting for a specific signal, waiting for a specific set of proteins to show up. And once that signal or that set of proteins shows up, then the cell proceeds through that checkpoint into the next phase of the cell cycle. We saw that one of the most critical cell checkpoints is something called the restriction point. It occurs in the G1 phase. If the cell goes past the restriction point, it's going to go through S phase, which means it's going to replicate all of its DNA. And because of this, it's committed to doing one of two things. Either going through cell division, going through the process of mitosis, or if something goes wrong during the process of mitosis, committing to going into cell death. So here the cell is committing to doing one of two different things. So there are other checkpoints in the cell cycle. We're only going to focus on this guy up here, this G1 checkpoint, also known as the restriction point. A lot of cancers have problems with the restriction point. In fact, one of the master regulators of the restriction point is a protein called E2F. Something like 60% of cancers have problems regulating this protein called E2F. So it's a really important checkpoint for health and everything else. So the idea is that if the cell receives signals, then it will proceed through the checkpoint. The gate opens up, and the cell proceeds, in this case that we're talking about the restriction point, from the G1 phase into the S phase. However, if those signals never come, the cell enters into an indefinite G1 phase that we call G0. So the cell's just going to sit there, and it's going to perform its normal cellular functions. You can take it out of G0 phase by giving it a lot of signal again to go back into G1. But most of our cells are in this sort of indefinite G0 phase, or just kind of doing things, processing, detoxifying, that type of stuff. At a molecular level, checkpoints are regulated by two broad classes of proteins. These broad classes of proteins, there's literally 50 or 60 of them specifically named. And in a class like cell and molecular, which is in 4th year, what you do is you start to learn how these things interact with each other and how they regulate the cell cycle. In general, though, you can break down these two groups of proteins into cyclins and kinases. So we've seen this word kinase before. This is a protein that goes and phosphorylates another protein. Cyclin is new. A cyclin is a protein that changes with the cell cycle, or it cycles with the cell cycle. What that means is that in certain periods of the cell cycle, the cyclin will be present, and then in other periods of the cell cycle, it goes away. So its presence or absence changes. Now what these cyclins do is, or I should back this up and say the kinases are relatively constant. They're kind of always there in the background throughout the entire cell cycle. So the cyclins are changing, but the kinases are relatively constant. Now the role of the cyclins are to activate the kinases. So the kinases are always there, but for the most part, they're off. They don't have the ability to go out and phosphorylate other proteins. What they're waiting for are cyclins to show up, because the role of these cyclins is to interact with these kinases when they're off and to activate them. And then what's going to happen is these kinases are going to go out and start phosphorylating things and push the cell through a checkpoint. So if we were to kind of draw this out really quick, it kind of looks like this. Draw sort of what's going on over time here. So this is time. I'll draw the cyclins in black pen here. The cyclins kind of do this. Let's say this is concentration. So here, the cyclins, what they do is they do this. So they oscillate or they change during the cell cycle. Kinases, on the other hand, they do that. They just flatten. So these kinases, we don't want them on the whole time. If they're there the whole time, we don't want them constantly phosphorylating things. What we want to do is we only want to activate them when a specific cyclin shows up. And the idea here is it kind of looks like this. So once a cyclin shows up, what it's going to do is it's going to interact with one of these kinases that turns the kinase on. And then that kinase is going to go out and it's going to add phosphate groups to other proteins. And then that gets you through the checkpoint. So cyclin turns on a kinase.
That's you.
The kinase is going to go out and phosphorylate other proteins. And once those proteins are phosphorylated, the cell moves through the checkpoint. All right, questions so far? Okay. These kinases, remember a kinase is a word that describes any protein whose role is to go out and phosphorylate something else. That's all it says. Now these proteins here, these are activated by cyclins. So they're only turned on when a cyclin shows up. And because of that, we give them a really specific name. We call these guys cyclin-dependent kinases. And what the community does, the cell cycle community or the cancer community or something like that, what they do is they abbreviate these guys CDKs. So if you're reading a paper and it says something like CDK6, they're talking about a cyclin-dependent kinase, number 6. Maybe the sixth one they discovered or something like that. So if you type in Google Scholar, which is an excellent place to look for primary literature, you just type in the word CDK, you'll get tens of thousands of hits in terms of people trying to understand what these do, how to manipulate them, how to put them back to baseline when they get screwed up, that kind of thing. Okay, so to put some perspective on this, in Cell and Molecular, and if you take my systems biology class, which is another 4th year class, we talk about this type of stuff. We actually spend a lot of time talking about the restriction point in my systems biology class because it's so integral to the cell cycle and integral to life. We go through the dynamics of it, we talk about some of these CDKs and how they work and that kind of thing. You might also talk about them in biotech, depending on who you get. Or any cancer class you take. Questions? Okay, so just flip over here. So we kind of talked about this last little part of the slide here, how we call these cyclins cyclin-dependent kinases because they rely on kinases in order to activate them. And then once these kinases or these CDKs are in their activated form, they go out and start activating other things inside the cell. Questions about cell cycle regulation? Again, we are like just glancing over the surface of this thing. All right. That's it for mitosis. Any questions about mitosis? All right. Anybody go to homecoming last night?
Oh, the pep rally?
Yeah, sorry,
Yeah.
that's what it is. Yeah, was it fun?
I got a shark.
That's what I wanted. They wouldn't give me one though.
Yeah.
Like, are you a student or? Maybe.
Yes. I'm a grad student.
Yeah, right. I can go with that. They sang out the bowl.
Whoa.
Well. Okay. I'm just messing with you.
No, it's okay.
All
I
right.
can do that.
Let's fast forward. What we're going to do is we're going to talk about meiosis here. Now, the key to this lecture, okay, we already talked about sort of what's going on with mitosis in terms of where chromosomes are moving. If you guys weren't in the last lecture, which looks about half you guys didn't come to last Thursday's lecture, I made a point to point out that there's a ton of words that start with the letter C in that last lecture. I strongly recommend you build a table and write down the definition of those words that start with C, because they all become really confusing when you hit the test, or in this case, the final exam. And the same thing goes with kinetochore. It sounds like it starts with a C. You might as well put it on your list. We looked at the components of how mitosis moves, like what happens in metaphase and prophase and all that kind of stuff. So we're not going to pay particular attention to that in this lecture because we've already seen it, and a lot of the steps are the same. So we'll point out some differences, but we're not going to go through it again. Instead, what we're going to do is we're going to count chromosomes in this lecture and get used to figuring out how the number of chromosomes changes in between cells at their different stages, both within mitosis and in meiosis. And what this helps with is understanding the difference between mitosis and meiosis in terms of what the end product is and what the overall goal is. So if you get pictures of people from the 70s, which apparently is what your textbook puts in, what you'll see here is that people in this image kind of look like, here's some parents, and their kids kind of look like a blending of traits from the parents, etc. And the idea is that we as a species, and a lot of other species do the same, we've decided in an evolutionary sense that sexual reproduction is the way to go for us. And sexual reproduction, what it leads to is variability in offspring. So you kind of sort of look like your mom, and you kind of sort of look like your dad, but you're certainly not a clone of them. You're kind of a blending of traits. And the idea here is that sexual reproduction, because we're mixing and matching traits, it might lead to new combinations of traits that, in a natural selection purpose, might make you be able to outcompete everybody else. Now, we really don't do that as like human population, but that doesn't mean that other animals like birds and rabbits and bears and whatever else, they still do this type of things. They're all competing for a limited amount of resources. Okay, so the goal of meiosis is to produce variation at the genetic level. And the idea is that that variation at the genetic level should translate to variation in the offspring. And the idea then, a really long time ago, natural selection would then act on that variation. Just as a complete side note, something really cool that came out last week just popped into my head. It turns out that everybody on this planet can trace their roots back to 8 women that all lived in modern-day Botswana. So we all share, well, I mean, there's a really good probability that at least one of you and I shared a really ancient common ancestor. In fact, there's probably a really good group of people that share the same common ancestor. So we all came from 8 women in Botswana. So I haven't read the article in terms of how they figured it out, but that's insane when you think about it. The fact that they're able to go back through thousands and thousands and thousands of years to come up with this. So if you're looking for something interesting to review, that's something cool. Okay, so we want to produce genetic variation. Remember, this is different from mitosis. The goal of mitosis is to produce clones. We don't like genetic variation in mitosis. Genetic variation into mitosis kind of sort of leads to disease states. Okay, so we've picked sexual reproduction. Other animals pick asexual reproduction. They'll use mitosis as a form of asexual reproduction. So things like bacteria do this. Here's an example for everybody who's in sort of like marine biology or something like that. It's called a hydra. It's kind of like an upside down jellyfish. These things are really, really, really small. And they go through asexual reproduction. And the idea here is that they're not really worried about generating a lot of variability in offspring. What they're relying on is just putting out a ton of offspring into the environment and hoping that some of those offspring make it. So here's sort of the parent hydra, and what it's doing is it's kind of sprouting off a little bud, and eventually this bud will break loose from the parent, it'll float in the current, it'll attach to a substrate like a rock or coral or maybe the ground, and then it'll become its own plant. The key thing here, though, is that this bud is exactly the same at a genetic level as the parent is. So there's no genetic variability here. For hydra's, asexual reproduction seems to work really well. So this is all driven through mitosis. So anytime you hear asexual reproduction, the vast majority of the time it's through mitosis. And again, you're not trying to generate variability. Questions so far? Okay, yeah.
So even if you go, like, even if it's through mitosis and asexual reproduction, is there still possibility for variation?
Yeah, so these guys will go through a different round. They will go through sexual reproduction, but it's not their main form of reproduction. They kind of have this strategy where they're like, okay, I'm going to make a bunch of clones of myself, hope that these survive, and then at some point when times are really good and there's lots of nutrients or lots of sunlight, whatever it is, then I'm going to go through sexual reproduction. Now, of course, there's always a chance that there's a mutation in the cell that initially arose to give rise to that bud. So, in theory, not all of those buds will be 100% genetically identical. There's always a chance that they pick up a mutation. Other questions? All right. So we're going to first sort of talk about humans here. Again, the goal of meiosis is to produce genetic variability. And with us, we have two parents that give rise to an offspring. And the idea is that even though you may be very similar to your brother or sister, realistically, you are very different at the genetic level. The chances of you getting an exact same DNA, exact same copies and everything else as your brother or sister are incredibly, incredibly low. The only exception to that is twins. And even in twins, there are what are called single nucleotide polymorphisms, so changes in single base pairs that might make them a little bit different. And there's also what are called epigenetic changes. These are changes not necessarily to DNA sequence, but to maybe how your DNA is folded in between twins. So there was an experiment where they took a twin, put the twin in space, kept the other twin on the ground. You guys have heard of this? And then looked at how their genetics changed when this guy went around the Earth as opposed to the guy that stayed on the planet. And apparently when you go around the Earth, not only do you age slower, but there are changes to your DNA that you don't see when you're on Earth. And they did this to try to figure out how missions to Mars would impact people's DNA structure, as well as their physiology and their thinking and that kind of thing. Okay, so just as like a really brief refresher on some words here that have to do with genetics, because now we're entering into genetics land. Heredity, this is you passing on traits to offspring. Variation are sort of differences in either phenotype, so the traits that an individual displays to the world, or genotype, the genes that they have inside their cells. And then genetics is the study of heredity and heretica. Genetics is basically the study of genes. And then genes is, for the purposes of this class, it's an area of DNA that encodes some type of trait, or encodes a protein. I won't ask you guys these definitions on the final exam. So I wouldn't worry too much about memorizing them. Okay, so the way that you go through, or the way that you produce variability of offspring is you use something called gametes. So these are sexual cells that are meant to produce offspring. And the idea is that when you fuse two gametes, you can make a zygote, which then turns into a fetus, which then turns into a human. And the fusing of gametes is what leads to genetic variability. For us, the two gametes that we have, males have sperm, females, on the other hand, have eggs. This is sort of a general rule throughout biological systems, but this can also vary tremendously in terms of what these cells actually look like and how they're used. For example, here's a fun fact you guys can drop in the club when you guys are 21 years of age or older. One crazy fact about fruit flies is that the sperm of a fruit fly is longer than a fruit fly itself. How the hell does that work, right? So what they do is they package up the sperm, they roll it into the smallest ball possible, and then they launch it out, and if you stretch out that sperm from end to end, it's longer than the entire fruit fly itself. Another interesting factoid about fruit flies has nothing to do with meiosis. If you give them a concentration of ethanol, where at the bottom of the tube they'll die because they've had too much ethanol, and at the top of the tube they don't really feel the effects, the fruit flies will go hang out in the area where they get sort of drunk but don't die. It's really bizarre. Okay.
They're responsible drinkers.
What's that?
They're responsible drinkers.
Right, they are. But it's crazy because if you look across the animal world, you can find examples of this behavior in lots of different animals. So for example, humans are unique in that we have a gene called alcohol dehydrogenase, which is a gene that's meant to detoxify alcohol.
Right.
Things like dogs and cats don't have this. That's why if you feed your dog or cat beer, it's really bad for them. It can kill them. So the reason why we think we have this gene is that our ancient ancestors, and I mean things like bonobos, like really ancient ancestors, what they probably did was they ate rotting fruit off the ground that had undergone fermentation, and then they got ethanol during that, and that sort of drove alcohol dehydrogenase to become prevalent in the population. But you can still go out in the field and see apes eating rotten fruit on purpose to try to change to get drunk, basically. So it's kind of one of this really weird thing that's prevalent throughout the animal kingdom. It doesn't really have any advantage. It's just something a lot of animals do. Okay, so off of this. So let's talk about what we look like in humans, sort of at the meiosis level. Okay, so I have some definitions up here first. So karyotyping, which we're going to see on the next slide, this is when you take all the chromosomes out of a cell. The cell's typically in M phase, specifically metaphase. And what you do is you line them up so they look really nice. And you end up getting pictures that look like this. So this is something called a karyotype. This is how most often you guys have probably seen chromosomes. I know that when I was in your shoes, this is how people always presented chromosomes to me. We're going to come back to this image in just a second so we can go through some of the morphology of these guys. The reason why karyotyping is important is it allows us to figure out how many copies of a chromosome and how many total chromosomes each cell has. So for us, we have a total of 46 chromosomes. We get 23 of these from mom, and we get 23 of these from dad. Chromosomes 1 through 22, these are non-sexual chromosomes. These are what we call autosomes, or autosomal chromosomes. So they don't encode genetic sex. On the other hand, we have two different sex chromosomes. These represent the 23rd pair. There's two different types. We have X's and we have Y's. And these determine the genetic sex of the individual. So if we look at this karyotype here, let's just focus on this image first, so on the right-hand side of this slide. All right, the chromosomes don't actually look nice kind of rainbow colors like this. This is either taken with a piece of software that looks for density, or they've been stained with something. Perhaps someone's looking for something specific in these chromosomes. Usually they're just kind of like shadows or black sort of against a white background. In any event, so we have 23 pairs. Okay, so here's our autosomes labeled 1 through 22. Here's the last pair of chromosomes right here. This is our X and Y chromosome. This individual, because they have a copy of X and a copy of Y, this individual would be a male at the genetic level. If this individual had two X's, they would be a female at the genetic level. All right, and the idea here is that people do this in order to look at chromosome number, because sometimes chromosome number affects sort of what the offspring does, or they try to do this in order to figure out if they discovered something new, how many chromosomes that new organism has, that type of thing. Okay, so while we're talking about this, this is a really good picture over here. So let's switch over to the left-hand side here. What you're seeing here, remember this is taken in metaphase. Have we gone through S phase and metaphase?
Mm-hmm.
Yeah, we have, right? We've been through S phase. So what does that mean we've done? Right, we've copied our DNA. We've gone through DNA replication, right? Okay. So what I want you to do is look at half. Okay, just look at this guy right here. What you're seeing here is a pair of sister chromatids. So these guys here are genetically identical to each other. And they're being attached through a centromere. So we've seen this before, right? We saw this from mitosis. These two guys over here, if you compare them, they're also sister chromatids because they're attached through a centromere. So for example, when we're looking at a karyotype over here on the right, this chromosome here actually represents two sister chromatids stuck together using a centromere. And this guy right next to it also represents two sister chromatids stuck together using a centromere. Okay, questions? That's going to come back. It's going to be important in a couple slides. Any questions? Okay. At this point, what we're going to do is we're going to start keeping track of the number of chromosomes that we see at each stage of the cell cycle towards going through meiosis. Okay? One of the key things in meiosis is that the gametes that we produce are something called haploid. Haploid is a word that we use to say they only have one copy of each chromosome. Because our cells are mitotic cells, the ones that go through mitosis, or what also known as our somatic cells. These guys have two copies of each chromosome, so we call these cells diploid. Now, if you're not us and you're another organism and you have 3 copies of chromosomes, you're triploid. If you're types of, especially like fruit and that kind of thing, that have 7 or 8 copies of chromosomes, these are things like heptaploid and octoploid. So, but just for humans, haploid means we only have 1 copy of each chromosome, and diploid means we have 2. The goal of meiosis is to make haploid cells. That are cells that only have 1 copy of each chromosome. Then what we do is we take 2 gametes and we fuse them together. We take the 23 chromosomes that are in one gamete and the 23 chromosomes that are in the other gamete. When we fuse them together, how many total chromosomes do we have in that cell? We have 46. That represents a diploid cell. Okay, so I want to go to this first here, so I'm going to skip one slide. So in humans, okay, what we do here is during meiosis, we start with cells that are diploid. And then what we're going to do is we're going to divide those cells until we get gametes that are haploid. So in humans, over here, we are diploid cells with 46 chromosomes. So the way that we can tell we're diploid, or that this is denoting diploid, see this little 2n down here equals, excuse me, that 2n, that 2 means you're getting 2 copies. If it said 3, it'd be 3 copies, 4 copies.
Mm.
So what we do in order to make gametes, remember we're trying to make gametes that only have 1 copy of each chromosome, is we're going to take diploid cells, We're going to put them through the process of meiosis. And at the end of meiosis, we will generate gametes that only have one copy of each chromosome. In this case, they have 23 chromosomes in them. The way that we denote that is we say n equals 23. So that n represents haploid, and our gametes here only have one copy of each chromosome, so they have 23 chromosomes. Questions so far?
Wait. So you start off like diploid, then you go haploid, and then you go back to diploid once it's like fertilized?
Absolutely.
Okay.
Yep, you're absolutely right. So the next step in this, what's going to happen is we're going to fuse two gametes. So sperm from dad, eggs from mom. What ends up happening is we take the 23 chromosomes from dad and the 23 chromosomes from mom, and we fuse them together to make something called a zygote. This zygote contains one set of chromosomes from mom, one set of chromosomes from dad. Therefore, the total amount of chromosomes we have is 46. And because we got one copy from mom and one copy from dad, we're now diploid. And then from this one cell, we grow into billions and billions and billions and billions of cells. And then we enter into this stage here. And then we make more gametes, and the cycle goes round and round and round and round. So basically, we start off as diploid cells. We make haploid cells, but then turn into diploid cells again. Questions? Does anybody have any questions with this? Yeah?
So are the haploid cells made just from like other diploid cells?
Yes. So basically, there's what inside your body. There's specific cells called germline cells. The goal of these germline cells is to exclusively produce gametes. And all those germline cells hang around in diploid. And after we put those germline cells through the process of meiosis, they become haploid cells. Other questions? All right. This guy right here, or this slide, is kind of key in terms of figuring out some of the terminology. Now we're going to add one more piece of terminology to our sister chromatid, or I guess two more pieces of terminology to sister chromatid, centromere, all that kind of stuff. This is meant to represent a simplified cell. So this is a cell that would not be representative of ours because it doesn't have 46 total chromosomes. Instead, what we can say from this cell up here is if you look at the top, in its diploid state, 2n equals 6. It means it has 6 total chromosomes. If this cell were to go through meiosis, how many chromosomes would its gametes have? It'd have 3, right? So here it exists in its diploid state. Gametes are trying to get to haploid state. So here, half of 6 would be 3. Everybody okay with that? How I got there? All right. So what they've done is they put a key on here. The red chromosomes are the chromosomes that come from mom. The blue chromosomes are the chromosomes that come from dad. Okay? What I want us to focus on here are the big chromosomes. So this guy here and this pair here. Now this is a cell that sits in something like metaphase. So it's gone through the process of cell division. It's gone through the process of S phase. These two chromosomes here, okay, if you focus on these big blue guys right here, they're attached to the centromere. Both of these chromosomes are genetically identical. Because of that, we call them sister chromatids. So this is no different than what we talked about when we talked about mitosis. If we focus on, again, the big chromosomes, but the chromosomes in red, These guys are attached through a centromere as well. Both of these guys are considered to be sister chromatids because they're genetically identical. Yep.
So because I remember from like the past slide, like are each of them like sister chromatids that are like held together by the centromere, like two?
Yep.
Each one of those tubes that
Yep.
is like in black outline has two.
Correct. So if you look at this sort of big blue pair right here,
Yeah.
what that pair standing alone is meant to represent is this guy right here,
Okay.
or one of these guys right here.
Okay.
All right. The confusing part to this that I'm going to add is this. If you take this pair of chromosomes and you compare it against these guys here, they're both big chromosomes in comparison to everybody else here. But they're not genetically identical. So if you take this guy here and compare it to this guy here, they're no longer genetically identical, but they're still the big chromosome. If we were to compare those two chromosomes against each other, we'd call them non-sister chromatids. So they're still the same chromosome, that they're the big chromosome, but they're no longer genetically identical because one came from mom and one came from dad. So we call them non-sister chromatids.
This is still in metaphase, correct? Right?
Yes, yeah.
Oh, okay. Yeah, that's fine.
Other questions? All right, everybody's good with that. So non-sister chromatids, they're the same chromosome in that they're either chromosome 1, 2, 3, 4, 5, 22, whatever it is. But they're non-sister because they're not genetically identical, meaning that one came from mom and one came from dad. If we get both of these pairs and we stack them together, and you're talking about both pairs together, so both pairs of this big chromosome, we call them a pair of homologous chromosomes. So homologous chromosomes, it would be like, again, if you're grouping this guy here together with this guy here. Or as the slide says, you're grouping these two sort of intermediate sized chromosomes together. So the two new pieces of terminology we're adding here, non-sister chromatids and a pair of homologous chromosomes. Questions? All right. Everybody's cool with homologous pair and non-sister chromatids versus sister chromatids. Everybody's cool? Everybody's cool? All right. Now let's look at the process of meiosis, okay? So a lot of the actual steps between mitosis and meiosis are very much the same. In that, in meiosis, you're still going to go through prophase, prometaphase, metaphase, anaphase, telophase, and a lot of the steps in each one of those phases are exactly the same. So kind of when you're studying mitosis, you're also studying stuff for meiosis. You just kind of have to look at the differences between meiosis and mitosis. One major difference in meiosis is that we go through cell division, meaning we go through prophase, metaphase, anaphase, telophase twice, whereas in mitosis we only went through all of that once. All right, at the end of meiosis, we're going to have 4 haploid cells. On the other hand, in mitosis, we were looking at having 2 genetically identical diploid cells. So I'll say that again. Meiosis at the end, we have 4 haploid cells. That's different than mitosis. Mitosis, we're looking for 2 genetically identical diploid cells. Okay. What we're going to do here is we're going to draw out differences between mitosis and meiosis in terms of sort of cell cycle and that kind of thing, okay? And we're going to come back to the slide, but I feel like drawing this out kind of helps interpret what's going on next. So just like other classes, what I'm going to do is I'm going to take these sheets, I'm going to put them up front here. So you want to take them as souvenirs. All right. So, final exam, what I'm going to do is I'm going to ask you questions like, if a cell started off in G1 phase with a diploid number of 10, how many chromosomes does it have in its haploid cells? Or how many chromosomes does it have after S phase? So what we're going to do is I'm going to show you some examples of this. And the idea here is it's going to help you understand sort of the differences between meiosis and mitosis in terms of how the numbers of chromosomes are moving. So first, we're going to draw mitosis. All right. So we'll pick a cell. We'll just say, okay, so in this cell type, it's somatic cells, meaning the cells that go through mitosis. They have 2n equals 16. This is a diploid number. What this means is that they have 2 copies of 8 chromosomes. All right. So when this cell is sitting around in its G1 phase, this cell has 16 total chromosomes. All right. Everybody good so far? After S phase, how many total chromosomes is this cell going to have? Close. It's going to have 32, right?
Oh, I don't know how to do math.
Yeah. So after S phase, remember what we're doing is we're duplicating all of our DNA. So we're going to have a cell here with twice as much DNA. So another way of saying this, to transition between G1 and S phase, we're going to multiply everything by 2. How many chromosomes does this thing have in G2 phase? All right, has it gone through cell division yet? Does it get rid of its chromosomes? No, so it's still going to have 32 total chromosomes here in G2 phase. Then what's going to happen, right, is that this cell is going to undergo mitosis, and it's going to take these 2 sets of chromosomes, split them evenly between 2 cells. Therefore, after M phase is over, you generate 2 cells with 16 chromosomes each. And then the cycle repeats itself, right? If there's one of these cells wanted to go through the process of mitosis again, just do this whole process again. So for us, our cells do this, but the numbers change where this is 46, and then a whopping 92, and then we go back to 46.
Mm-hmm.
Questions? Okay. Everybody have this down okay?
Yep.
All right. Let's look at what happens. We're going to take the same cell, but we're going to put it through mitosis, or excuse me, meiosis. So over here is meiosis. Again, we're dealing with a cell whose diploid number is 16.
Mm-hmm.
So when this cell is sitting in G1 phase, it has a total of 16 chromosomes. what's going to happen? It's going to go through S phase. And in S phase, what we do is we double the amount of chromosomes. Cells undergoing meiosis still have G2 phase. We're going to have the same number of chromosomes here, which is 32. And then what the cell is going to do is it's going to go through cell division. Now, there's some little tiny differences in between mitosis and meiosis in terms of cell division, but the overall principle here is the same. The first thing that this guy does is it goes through a process called meiosis number 1, where it's going to go through cell division, and it's going to create 2 cells with 16 chromosomes each. Does the cell stop here? No, right? It can't, because it has to generate haploid cells. And the haploid number for 16 is what? 8. Okay, so what's going to happen here is the cell is going to go through another round of cell division. And this other round of cell division, we call this meiosis number, or meiosis 2. Each one of these cells is going to go through cell division. And what we're going to end with here are 4 cells that have 8 chromosomes in them. Or another way of saying this, 4 haploid cells. Yep.
So do those for meiosis 2, do they go through all the the G1, S, and G2 phases again?
So they won't. What they'll do is they'll basically hit telophase and cytokinesis of the first meiosis. And then almost immediately, they go through another round of cell division. So they'll go right into prophase, prometaphase, that kind of thing. So that's a really good point. At this step here, they don't go through G1 and S and G2 again. They just go right into the next meiosis. Yep?
What type of, is there anything in the body that goes through mitosis?
So the vast majority of your cells go through mitosis. So any cell that is not a germline cell is going to go through mitosis.
So meiosis is only for
germline cells or cells involved in sexual reproduction.
Okay.
So it's like a minority of cells in your body.
Okay. Thank you.
You're welcome. Other questions? So, if I put 2n equals 24 at the top here, all you have to do is just adjust the numbers throughout this whole thing. If 2n equals 24, what's the number of chromosomes down here?
It's 12.
It's 12, right? So remember, if 2n, meaning diploid, is 24, you're trying to generate haploid cells. So all you do is take that value up here. So if this was 24, just divide it by 2. That gives you the haploid number. That's the number of chromosomes that your gamete should have. So in that case, it would be 12. Questions? All right. Everybody okay with this? Okay. So if you didn't get this down, or you want to come up and take a picture, again, I'll put it kind of up here. Okay. So this slide right here is pretty much showing us what we just did. Okay? But it's only showing it for one set of chromosomes. So with the paper here, we were following, 16 chromosomes as they move through this process. Here, they're just looking at one. So what they're saying here is, here's one chromosome that you got from dad. Here's the other chromosome you got from mom. What's going to happen is in S phase, we're going to replicate these things. Right? So you have ones you got from dad stuck together with a centromere. You got the ones that you got from mom stuck together with a centromere. During the first round of meiosis, we're going to split those cells into, or split these chromosomes into 2 different cells. And And then in meiosis number 2, we're going to split the chromosomes in each one of these cells into another 2 cells. And at the end of the day here, you're left with 4 haploid cells. So in this case here, this cell, it would be 2n equals 2. So you'd have 4 here, and then 2, and then 1. Yep.
Alright, I mean, I learned this like way back in high school, so I probably don't remember like exactly, but like, wasn't it that like, because like here it's just showing that like, okay, like like from the father's side it goes into like 2 cells, and then the mother's side goes from 2 cells. There's no like.
So this is I don't like this slide a lot, which is why I do this, because the slide's misleading in exactly what you're pointing at.
There's no cross, right?
There's no crossing over
Okay.
here. So it looks as though you're just creating like dad cells and mom cells from this, which is complete BS. What we're going to see is that these chromosomes, non-sister chromatids, start exchanging pieces of DNA in the next couple of slides.
Okay, I'm not going
Yeah,
crazy.
so you're right. No, you're absolutely right. Okay, so we went through, before I move forward, any questions? So we went through some of these definitions already, where we said sister chromatids. These are genetically identical cells that are attached together using a centromere. And a homologous pair consists of 2 sister chromatids from dad and 2 sister chromatids from mom. They get really kind of close to each other. They're the same chromosome in that they're either chromosome 1, 2, 3, 20, whatever it is. All right. Now what makes non-sister chromatids is that there's variability in their DNA sequence. Because remember, you got one from mom and one from dad, so they're not going to be identical. What we're really worried about in terms of what's different between them is something called alleles. So I know you guys just did the fruit fly Mendelian genetics lab, right?
Yes.
How was that?
Yeah.
Okay. Did anybody else find that lab a little confusing?
Yes. Yes.
Okay, cool. Because what we're going to do is I'm going to show you guys Mendelian genetics again next week. I'm going to simplify it a little bit for you guys. Okay. In any event, you guys learned about this terminology called an allele, right?
Yeah.
So really what we're worried about is just alleles on these chromosomes that make them different. So it's things like hair color, eye color, that kind of thing. So simple traits like that. Now, in reality, humans aren't that simple. There are very few traits that just encode one thing. It's usually multiple traits coming together. But an allele is just sort of a variation of a gene. So what makes non-sister chromatids different is they have different alleles on them. Okay, before I move forward, are there any questions about terminology with respect to homologous pairs, non-sister chromatids, sister chromatids, diploid, haploid, that kind of stuff? Mm-hmm.
What's the difference? This is like going a little bit back. What's the difference between like the gene, like the hereditary, and like an allele? Because like I was trying to understand that in like lab, and like for the life of me, I couldn't get it.
Difference between a gene and an allele. That's a really good question. So, when people talk about alleles, they're not looking at all the building blocks that make up a gene. Or they're not looking at the physiology of the gene. What they're just saying is that here's a chunk of DNA. We don't care what it looks like. It just encodes a trait. And we call that an allele. A gene, on the other hand, is more of a, when you talk about a gene, you're more worried about the physiology of what that looks like. What does the promoter look like? What does the terminator look like? What regulatory elements are there upstream of it? That type of thing. So one of them is more of a, like a bigger picture, which is an allele. The other one's more of a molecular question. At the end of the day, they kind of mean the same thing.
Oh, okay.
Yeah, it's just depending on what level you're looking at.
Okay, thanks.
Other questions? All right, so because because we've seen the steps in mitosis, what I don't want to do is go through all of the steps in meiosis again. What I want to do is I want to point out some important differences between mitosis and meiosis. So here we're going to look at the difference, some of the unique features of meiosis. So again, when you're studying the steps in mitosis, you're really kind of studying the steps in meiosis at the same time. The first key difference with mitosis and meiosis, we talked about this already. In mitosis, the process of cell division, meaning prophase, prometaphase, anaphase, all that kind of stuff, happens once. In meiosis, it happens twice. So the cell's going to go through prophase, metaphase, anaphase, telophase, and then it's going to go through the same thing again, prophase, metaphase, anaphase, telophase, immediately following that. That's meiosis. Because of that, what we do is we say the first time the cell goes through prophase in meiosis, we call it prophase 1, and the second time it goes through it, we call it prophase 2. So what the cell is going to do is it's going to go through prophase 1, metaphase 1, anaphase 1, telophase 1, and then it's going to go through prophase 2, metaphase 2, etc., etc., etc. So that's the first major difference here. The next major difference is what happens leading up to the process of cell division, or meiosis number 1. So at the end of G2 phase, or kind of in the beginning of prophase 1, what's going to happen is you're going to have a unique event in meiosis called crossing over. And in crossing over, what's going to happen is you're going to exchange regions of DNA in between non-sister chromatids. So the key thing here is this crossing over, or this genetic exchange, occurs between non-sister chromatids. We don't want to have this happen between sister chromatids, because remember, sister chromatids are genetically identical to each other, so you'd just be flipping pieces that are exactly the same. But if we do this between non-sister chromatids, now we're generating genetic variability. We're reshuffling genes. This crossing over event, this is where we get variability in our offspring. So they're not clones of the parents. Okay, so the way that crossing over happens, we're going to get homologous pairs that link up together. And these homologous pairs, what they do is they form a structure called the synaptonemal complex. So if you want to sound really smart around your friends, just use that one. You can use it inappropriately too in like a non-science context, especially if there's like, you know, you're hanging around non-science friends. Just throw it out there. It sounds really smart.
Say it again.
Synaptonemal complex. Yeah. You know, maybe you're doing like, I don't know, you're building stuff from IKEA, right? And you're just like, oh, you know, step 6 here says we have to put this into a synaptonemal complex. And you just sound 1,000 times smarter. They probably just mean, you know, connect two things together with a screwdriver. But. All right, now this, when they enter this, they're entering into a state called synapsis. And basically what they're going to do in synapsis is they're going to undergo this process called crossing over. So you're kind of looking at 3 things here. The synaptonemal complex is a structure. Synapsis is a state. And crossing over is what occurs. So again, crossing over, we're going to exchange pieces of DNA between non-sister chromatids. And this leads to genetic variability. All right. So I'm going to zoom in. On this image right here, so you guys can see it nice and big. To go with our color scheme, okay, so the color scheme is consistent with everything we've seen. The blue guys here are what we got from Dad, the red guys here are what we got from Mom. These two blue guys here represent sister chromatids. They're connected together via centromere. They're genetically identical. These two red guys right here, they're connected via centromere. They are genetically identical. They are sister chromatids. These guys here are undergoing crossing over. And what you're seeing here is you're seeing that this sister chromatid that you got from dad is exchanging DNA with this sister chromatid you got from mom. But because they're not genetically identical, what you're seeing here is you're seeing crossing over occurring in between non-sister chromatids. Or the red chromosomes and the blue chromosomes are exchanging pieces of DNA. Because they're non-sister chromatids, you're generating genetic variability here. So here is the same thing occurring here. You're seeing lots of crossing over in this guy here. These guys are also undergoing crossing over down here. So if you took these chromosomes, this is a homologous pair. The blue guys together are sister chromatids. And if you compare a red and a blue together, they're non-sister chromatids. Questions?
This is 1 or 2?
This is right at the start of prophase number 1, or kind of at the end of G2 phase. Other questions? All right, the last thing we're going to do in this lecture is add one more word that starts with the letter C to our list of crazy long words that start with the letter C that have to do with cell cycle. The word that we're going to add is chiasmata. Chiasmata are locations where crossing over is occurring, or locations where you're exchanging DNA in between non-sister chromatids.
Wait, so it's what?
Chiasmata.
No, but the definition.
Oh,
sorry.
areas where crossing the non-sister chromatids are undergoing crossing over, where they're exchanging DNA. Okay, questions? All right, so we are going to end here. What we're going to do, not Thursday, because Thursday we have tests, don't forget. What we're going to do is we're going to wrap up this lecture next Tuesday. And then what we're going to do is we're going to talk about Mendelian genetics. I'm going to try to simplify what they taught you guys in lab a little bit. See you guys on Thursday.
Test is as usual, right? At 8?
Sorry?
The test is at 8.
Yep, same, yeah, basically it's just like all our other tests.